next speaker is Selene Fernández. Selene, are you there, ready for
Hello, I'm here.
Hello. So I'll share my screen. Selene is from Mexico, Selene Fernández, and she works at the Advanced Genomics Unit, Langevio, Clinvest in Irapuato, pronounced Mexico, and her laboratory studies the evolution of long non-coding RNA. Today we are with non-coding RNAs in animals and plants, and develops bioinformatic tools that help predict their functions. She did her doctoral and postdoctoral studies at the University of Queensland, Australia, and she's a graduate from the Bachelor of Genomic Science of UNAM. It's National University of Mexico, UNAM?
Yes.
She's a founding member of Mass Science for Mexico and the Mexican Network of Bioinformatics.
Yeah, great. Okay. Thank you so much for the invitation. I am really thrilled to be participating in this. I think it's an amazing initiative. To be honest, I haven't been able to watch all the talks. I have a little one that I'm taking care of. We're working all from home, so it has been a bit difficult, but I try to binge watch at night as much as possible. So thank you all for tuning in to listen to my talk. So I will be talking about some work we recently published about splicing conservation of plant non-encoding RNAs. And I'm glad for those of you that were in the track before, we have heard a bit of non-coding RNAs. I will first introduce myself. So I work at Langevio in Irapuato, and this is, our new name is the Advanced Genomics Unit, Unidad Genómica Avanzada, and we are placed like right in the center of the country. And my lab works particularly on the study of non-coding RNAs, of different aspects of non-coding RNAs, including the identification, functional association, structural inference, evolutionary conservation, and finally the functional characterization of these molecules. I will be focusing today on the evolutionary conservation aspect. And well, you heard already about non-coding RNAs. So these are vaguely defined as RNA molecules longer than 200 nucleotides that lack protein coding potential. And they structurally resemble mRNAs, but they lack sequence conservation and are expressed at low levels. And they evolve really rapidly, which makes them really interesting from an evolutionary perspective. And what's also important is that the function of most of these molecules remains to be characterized. So long non-coding RNAs are prevalent in eukaryotes. This is a bit, I guess, not so new graphic, but it's quite, I like it because it's very clear the point that it's making. So in different species, including plants and animals, and also yeast, you find non-coding RNAs in different levels, long non-coding RNAs. So in pink, you see the number of proteins, and in blue, you see the number of long non-coding RNAs. And as you can see, the number of proteins remains relatively constant, and they're quite conserved, while the long non-coding RNAs vary a lot, and they disappear quickly once you go further back in time. And why are long non-coding RNAs interesting? We have found that mostly others have found that long non-coding RNAs participate in the regulation of transcription at different levels. A lot of their functions, and this has been touched upon before, is through changes to chromatin structure. And by establishing and removing epigenetic modifications through the recruitment of chromatin modifying proteins. And they also participate in establishing chromatin structure just via their transcription. And they serve also as repositories for proteins. So this thing called protein scaffolding, where subcompartments of the nucleus are actually put together by the appearance and disappearance of long non-coding RNA molecules. So one of the big conundrums that exist is that when we do things like de novo transcriptome analysis, or even just regular transcriptome analysis, we find lots of long non-coding RNAs. And there has been a lot of debate of whether most of them are functional or not. And we think we have an approach to this. So how do we identify long RNAs that are functional? And we define function, and this is important because it can vary a lot, surprisingly. A biological function is the reason why an object or process occurs in a biological system. And it implies a benefit to the presence of a particular molecule to these biological systems in a particular temporal or spatial context. And molecules with biological functions are maintained to a process of natural selection. So why is this important? I mean, it might seem pretty obvious to people who work with proteins, but long RNAs don't have the protein preserving the open reading frame constraint, which means they change very rapidly in sequence. However, it has been shown that many of the sequence, so for example, in this graphic, you can see that the green regions are highly conserved regions, and they're not all essential. While other regions, so for example, in exist, which is long RNAs that works in doses compensation in mammals, the low conservation region is actually essential, while the high conservation region is dispensable. So this makes it really tricky to study in terms of conservation, but we'll talk about a few approaches we can use. So what we try to use mostly is evolution. So just get informed by evolutionary signatures that might differ from those that are commonly used for protein coding genes. And I just wanna, you know, this, probably many of you know this quote, that nothing in biology makes sense except in the light of evolution. So how do we use this light? Well, we assume that long RNA sequences that have acquired function must be conserved. However, their evolutionary dynamics might differ from those that we generally studied. This is because most of their functions are presumed to be through their structure, and their structure might not be radically affected by changes as long as these changes in sequence, as long as these changes are compensated and maintain the pairings that preserve the structure, like the base pairings. So the different types of conservation that has been discussed, this is from this review from Igor Yulitsky in 2016, is if you have two species, we can have conservation of splice sites. We can also have sequence motif conservation. There are some long RNAs that are very highly conserved, but there are few. And we can also have conservation by synteny. So actually we know the long RNAs have the same origin, but they have changed radically structure, splicing, and sequence. And we can also have structural conservation. This is that even though the two sequences might not be the same and they might not even be processed in the same way, they maintain a similar structure once they are transcribed. And there have been some previous studies that have hinted at structural conservation. So up to 40, this study from Smith and collaborators found that 14% of the human genome has signatures of conservation at the RNA structure level. And just to remind you, less than 3% of the human genome actually codes for proteins, which means that a lot of that conservation is outside of protein coding regions. And a similar study remains to be carried out in other organisms. We're actually working on this in plants. We have finished it, but we are in the process of writing it up. And there have been a few studies tackling this question in animals. So for example, in this study in 2014, 2,500 long RNAs were found to be highly conserved, and 400 long RNAs were found to be over 300 million years old. However, this is very few if you compare it to actual protein coding genes. And this is another important reference study also from the laboratory of Igor Yulitsky. And what they did is they took a bunch of transcriptomes from different animals, and the idea was to see if the characteristics of non-coding RNAs were similar in all the species, if the transcriptomes were carried out in a very similar manner. So what they found is that indeed, long-encoding RNAs in all or most species, there were some outliers as usually in biological studies, but they found that they have lower number of exons than protein coding genes, and they were also shorter, and their maximal expression was also lower in general than protein coding genes. So just finally, as another piece of information, there have been a previous study in vertebrates, looking at conservation of splice sites. And what they found, and this is work from Nietzsche and from Peter Stadler's lab, they found that the conservation of splice sites was greater than the conservation of sequence in vertebrate long non-coding RNAs. So that takes us to what I actually want to talk today. So this is work primarily done by my student, a PhD student who's due to graduate this year, Jose Antonio Corona Gomez, and he's looking for postdoc if you're interested in a good bioinformatician. So just to provide a bit more context, there are a few non-coding RNAs for those people that don't work in plants. One of the best known examples of long non-coding RNAs in plants is cool air. And there is also a number of long non-coding RNAs that arise from this locus, the flowering locus C, which is FLC in Arabidopsis thaliana. And what's interesting about this long non-coding RNA is that it triggers an epigenetic switch mediated by polycomb between opposing chromatin states. So it basically controls flowering response to cold. I'm not gonna go too much in detail, but I'm happy to talk about it later if you are interested. But the main thing is that what has been found is that the structure of cool air is conserved between Brassicaceae. And there have been a few other attempts to assess conservation of long non-coding RNAs in plants, and particularly in Brassicaceae, when it was looked in this study from Mohammadin and colleagues in 2015, they only found 15, literally 15 long non-coding RNAs that were conserved by position or at the sequence level. There was another recent study from Nelson and collaborators where they actually look a bit more thoroughly and used genome alignments to guide their search. And they did find, so in yellow, you have protein coding loci, and they found they were more conserved. And in green, you have long non-coding lncRNA loci. So lncRNA stands for long intergenic non-coding RNA, which are the ones we generally study. So the signals of conservation are not clouded with the conservation you might find from protein coding genes. So they indeed found that they're lost more rapidly than protein coding genes, which is expected, but they were lost less rapidly than intergenic loci, suggesting there is some selection. And so this is the question we were asking: is there evidence for splice site conservation in plant long non-coding RNAs? And to do this, we generated a new very detailed genomic alignment of 16 species of Brassicaceae using a software called Cactus. And what's nice about this software is that you can actually change the reference species very easily from the particular format it uses. And we use both model plant species and domesticated species. And so we chose Brassicaceae because there's a lot of genomes. We only use 16, there's 33 genomes available. It has a robust phylogeny, small genomes, lots of genetic information, and is the main plant model organism with lots of genomic information and also reverse genetics. And we were interested as well, like from the evolutionary perspective, in domesticated species because it's a very, very strong selective pressure and for particular traits that we can track. So we can actually see if any long non-coding RNAs are associated with these traits, and if we can see the selective pressure in the actual genomic sequence. And additionally, to do this, we took a separate set of long non-coding RNAs that we had generated in the lab. So basically what we found is that the reference sets for long non-coding RNAs in plants, in particular in Arabidopsis, they didn't have as many spliced long non-coding RNAs, and this was at odds with what we have found previously. And it's because there hasn't been many recent re-annotations of these long non-coding RNA. So basically we supplemented our annotation with this data that was generated by my student, Irving Garcia, who just started his PhD, and this was his master's work. And he found a number of long non-coding RNAs, but the main thing is that we found 173, which were spliced and we used for this study. So we used the annotations from Araport, which is the main annotation, one of the main annotation sets for Arabidopsis thaliana. And we also use our own annotation. And here we're just comparing positional conservation in the genomic alignment. So it's literally just cutting up the alignment and seeing how much of these elements you're able to retrieve. And we found similar results to what Nelson and Al found. So obviously, well, as expected, coding genes were much more highly conserved than long intergenic non-coding RNA. So this is the set from our report, and this is our own set. And we found a similar trend, and we lose much more rapidly transposable elements. And we also studied other elements, but it's a very simple analysis. We just wanted to see if we were able to recover what had been reported previously. But what we really were interested in was in splice site conservation. So what we did for this is we used a number of transcriptomes from Arabidopsis thaliana and Brassica oleracea. And these were really deep stranded with replicated transcriptomes. And these were aligned using STAR to the transcriptome, sorry, to the reference genome. And we were particularly interested in good alignment to splice sites, because that's what we used to call splice site beyond just the annotation. So just to clarify a bit, like the annotations in Arabidopsis thaliana are really good, but the annotations of some of the other species we were using are not as good. So we wanted to see how well we were able to recover splice sites that might not be in the annotation, but that are true splice sites. So to do this, we generated models of the donor and acceptor site. And these were used in a program called MaxEntScan, which basically gives you a score, an entropy score that tells you how good or, well, how well the splice site adjusts to your expectation. So just in these graphs in the left-hand side, you see in pink, the Arabidopsis thaliana transcriptome, and in green, the Brassica oleracea transcriptome. And in blue, you see orthologous splices in the other species that were called just from looking at the orthologous site in the rest of the alignment and the rest of the species. And this is the maximum entropy score. And on the right-hand side, you see the same for the acceptor side. So this is the donor side, and this is the right-hand side is the acceptor side. And just to make it a bit more simple, let's say, so the pink and green tracks were real splicing sites in transcriptome, in next generation sequencing data, and the yellow side, the blue sites were actually predicted sites. So we wanted to see how good or bad our predictions were. And we found that for real sites, they all had a positive maximum entropy score. So what we did is we only took those predicted sites that had a positive maximum entropy score and discarded those that were negative. And with this, we generated a splicing conservation map for all long non-coding and also coding genes in Arabidopsis thaliana and all these species. So basically what you see here, this is one example. This is a new long non-coding RNA that is not annotated in Araport 11. And we know it's expressed preferentially in leaves, but we don't really know what it does, and we're working on characterizing that. And what you see here is that the score in each of these species for each of the splice sites. So basically, when you have a very positive score, this is more likely to be a very conserved site. So we measured this across the whole genome, and what we found is that 65% of spliced Arabidopsis thaliana long intergenic non-coding RNAs are conserved in another Brassica species. And so we found 112 lncRNAs, spliced lncRNAs that have conservation in other Brassicas. So the divergence range between the species we chose is approximately 53 million years. And this is actually greater than current estimates for plan for conservation of sequence of long non-coding RNAs in plants. And however, we found that this is substantially lower than what was previously reported in placental mammals, which diverge approximately 100 million years between them, where they found around 70% of conservation. So basically, we have a similar observation in terms of having more splice site conservation than sequence conservation. However, we see a much more rapid loss of splice site conservation in plants, well, at least in Brassica than in mammals. And there are a number of confounding factors, which I have touched on, which here are poor annotation, a very small reference genome. So everything was done in reference to Arabidopsis thaliana. And it also has very few spliced intergenic non-coding RNAs. And this is not always the case for other plants. So in summary, what I have shown you is that most long non-coding intergenic long non-coding RNAs have splicing conservation, at least in another Brassica species. And we basically find that there is more conservation of splice sites than sequence as observed in animals, but this is much less than what has been found in animals. Like it's just, there is a faster loss of conservation in plants. And we would really like to do another study in a group that doesn't have this drastic genomic reduction, and also to further support this with additional transcriptomic data. So with that, I just want to thank the people who participated in the study, Jose Antonio Corona Gomez, Irving Jair Garcia Lopez, and we did this in collaboration with Professor Peter Stadler from the University of Leipzig, and our funding agency. So I particularly wanted to mention the For Women in Science program that has really propelled my career. And Norma Paniego previously mentioned the Cabana project, so please do check it out. The website is cabana.online. Literally, with that you can just get information about the different things we're trying to get going and move. We did a lot of in-person training, and now we're doing trying to move all of this into online training and to the organizers. And this is my lab. So just, yeah, that's Jose Antonio and Irving and Peter Stadler, and this is us nowadays, just we're celebrating the publication of this work. Thank you very much for your attention.
Thank you very much, Selene. Great talk, amazing work. So at the end, the long intergenic non-coding RNA are not conserved at all.
No, they are, but what's interesting is that they there are a few examples that show that you don't need sequence conservation to preserve function. And that's what's really intriguing. Another fact that's very, very intriguing is that, for example, in phenomena such as dosage compensation, they have a rise independently. So totally independent long non-coding RNAs participate in either activation or repression of a full chromosome. So it appears that this is a very rapid way of generating new epigenetic controllers.
Yeah, very, very, very difficult topic to be in. So do we have any other any question somewhere in the Q&A? No open questions. We have no more questions. So we thank you very, very much, Selene, and hope you enjoy the meeting.
Okay, thank you.
Thank you very much.